Hi guys, I thought I'd put together a short little tutorial so that when you go to do exercises 8 and 9 and 18, you had some sense of sort of the instructions that I gave the students last Tuesday. So for exercise 8, aseptic technique, what you're learning here is how to make some transfers from broths to broths, from broths to slants, from slants to slants, and from plates to slants. So you're going to have to make sure you carefully read over exercise 8. But I wanted to give you a couple bits of information. The first of which is I'm going to be sending you or posting on the Blackboard course shell this Word document that does a nice job, I think, talking about how to operate the Bunsen burner. So please make sure you watch this carefully, or maybe bring your tablet into lab on Wednesday when you come to do this lab, so that you can play this maybe and watch it as you actually manipulate the burner. Not that you have to do that, but that would be something you could consider doing. But you definitely need to watch this. All of the Bunsen burners that we used on Tuesday have been adjusted so that when you light them, they should be at the proper setting with the light blue cone in the center of the larger flame, and that's the hottest part of the flame, is at the top of that light blue cone, and that's what you want to achieve when you go to adjust the burner, and they talk about that in the video. But again, all the burners that we used on Tuesday should be all adjusted, so it's just a matter really of lighting them and they should be ready to go. But if they're not, you can watch this video and they talk about how to adjust it so that you get this hot inner blue cone within the flame. So please watch that. And then as we get into exercise 8 specifically, the first thing that they talk about is how to make a transfer from one broth to another broth. Now, you know what broths are. Those are the liquid types of media. And I'm going to, again, share a screen that I hope has some of these diagrams on it that I would have you reference there in your lab book. And this is not very clear. Why isn't this clear? That's better now. Okay, so this is figure 8.1 that talks about how to begin making a transfer from one broth to another broth. So Deb's going to have the cultures laying out there in the lab for you, and you're going to get an E. coli broth, and you're going to get a sterile TSB broth from the rack, and you're going to make a transfer. The first thing that you're going to do when you get your E. coli broth is you're going to mix the broth to agitate the cells to evenly distribute them throughout the broth. And on step 2 here of the diagram, they're showing the person shaking the tube. I don't know that that's necessarily a good way to do that. What I would suggest you do, and let's pretend that my pen is the test tube that the broth is in. I don't have anything comparable to use here better than this. So let's just again go with it. What you're going to do is you're going to take the test tube, you're going to put it between the palms of your hand, and you're going to just rub it back and forth to sort of agitate the contents of that E. coli broth, to mix the contents, because sometimes you get a sediment at the bottom. So you got to make sure you mix it. Another thing you could do is you could just tap the test tube against, say, your index finger of your opposite hand, and that would create a little vortex, and that will mix the contents as well. So either method is fine. What you don't want to do is shake it like this. Don't mix it that way, because when you go to take the cap off, it's going to, come down the sides of the tube. You're going to have bacteria all over the place, and we don't want any of that to contend with, right? Okay, so the next thing you're going to do is you're going to sterilize your loop, and they show here how to go in. Again, keeping in mind that the top of the light blue cone is the hottest part of the flame. So stick the tip of the loop into that upper part of the blue flame, just above it actually, and extend it all the way down into the flame, this initial part of the loop, until this all glows orange. And then as you pull it out, it will cool. It will take probably 10-15 seconds to cool, before you can go in and get some inoculum, some E. coli broth. So while that's cooling, you can grab your E. coli, and what you're going to do is you're going to use the pinky finger of your dominant hand, and you're going to grab the cap, the black cap of the tube, like they show here, and with the opposite hand, the non-dominant hand, in this case it'd be my left hand, I'm going to unscrew the cap. So the cap stays in my pinky finger, but I'm turning the test tube. Okay, so that's going to obviously allow you to take the cap off. Then you pass the mouth of the tube through the flame a couple times. And then your loop will be sufficiently cool that you can go down in, grab some inoculum, pull it out. You're going to reflame the mouth of the tube, and then you're going to insert it and then twist the test tube with your left hand, your non-dominant hand, while holding the cap stationary until you feel resistance, and then you can put the test tube in the rack. So now you've got the E. coli broth, a little bit of it, on your loop. You're now ready to make the transfer into the sterile TSB tube. So what you're going to do is you're going to get the sterile TSB. You're going to put the cap in the pinky finger of your dominant hand. You're going to unscrew the cap. You will pass the mouth of the sterile TSB through the flame a couple times, and then you'll inoculate it with the loop. Pull the loop out, flame the mouth by passing it through the flame a couple times, and then you're going to recap it, and then this is eventually going to be incubated, your inoculated TSB. What you have to do at the very last step here is to sterilize your loop, because there could still be bacteria on it. So you want to gently insert it into the upper edge of that light blue inner cone of flame until this whole thing glows orange. So you've just made your first transfer from a broth to a broth. Now you might wonder, why are we flaming both the mouth of the sterile TSB as well as the mouth of the E. coli broth? The reason that you heat this tube is to prevent any airborne bacteria from falling into it as you begin to make the actual transfer. Because remember, anytime a cap is off a tube or a lid is off a plate, there's potential risk for contamination. Things can fall, bacteria can fall from the air into that test tube of sterile broth or into our E. coli broth, or whatever the case may be, whatever media you're using. So you always want to be minimizing the time any lid or cap is off. So back to why we heat this. When you heat this and you pull it out of the flame, there's still heat emanating from the mouth. You can't see that, but think of a car in the summertime. You know, you can see the heat rising off the dark paint of the hood of the car, right? I'm just saying, you know, it's a hot summer day. The car's not on. You can just see the heat coming off the car or coming off the highway, right? So there is a vortex of air coming off that cooling mouth of the tube. And so any bacteria, let's say, let's pretend my arrow here is a bacterium in the air. This bacterium might want to, you know, fall down into that tube, but once it gets down toward the mouth, the vortex of cooled air is going to carry that off to the side so it can't fall down inside the tube. So it's kind of interesting why they do that, and at least the reason behind heating the mouth of the tube before and after you go in with your loop. So make sure you are following those steps. That's the transfer from a broth to a broth. Then you're going to be making a transfer from a slant to a slant. And Deb will have some slants, I think, with E. coli on them. You'll see the white snake-like growth of the bacteria on the surface of the agar slant. And then you're going to also be getting a sterile slant from the test tube that you'll have a rack of those. And basically, you're going to use the loop again. You're going to begin by sterilizing the loop. You're going to grab the cap of the E. coli slant and remove it like we've talked about, using the pinky finger of your dominant hand, unscrewing the test tube with your non-dominant hand, passing the mouth of the slant containing the E. coli through the flame a couple times. And then you're going to go in with the loop and just grab a little bit of inoculum, a little bit of bacteria that's going to be on the surface of that slant. You don't need to go down deep into the slant at all. Just go down as far as you need to go to grab a little bit on the tip of the loop. And that loop does not have to be covered with white growth, okay? Just grab a little bit of inoculum and then remove the loop. Then what you'll do is you'll reflame the mouth of the E. coli slant, place the cap back on, right? Then you'll get your sterile TSA slant that you've labeled. Don't forget to label these tubes, by the way. Name, date, what it is you're putting into it. You will remove the cap of the sterile slant. You will flame the mouth. Then you will go in with your loop. I'm sorry that this is blurry. I'm not sure why this is, but anyway, it's on page 67 of your lab book. You go in with your loop just above the bottom of the slant. You're going to see a little bit of moisture down there due to the condensation that's occurred. Don't go down into the liquid. Just go just above the bottom of the tube where the slant actually begins to form, and then begin to make a zigzag with your loop working toward the mouth. Hold the loop in such a way that it doesn't dig into the agar. Okay, but try to recreate basically what Deb did when she did her slants. So look at her E. coli slants. You're trying to do the same thing and use that entire surface of the slant as you zigzag your way toward the surface of the tube, toward the top of the tube. Once you're done doing your zigzags, you're going to flame the mouth, you're going to recap the tube, and then of course, don't forget at the very end, sterilize your loop again. Congratulations, you've just done a slant-to-slant transfer. The last thing we're going to do in exercise 8 is a transfer from a plate to a slant. So Deb's going to have some plates back there with E. coli on them. And what you're going to do is simply get your loop, sterilize it. You're going to gently lift the lid off the plate, minimizing the time the lid is off, because again, any potential opening allows for possible contamination. So just lift it up a little bit to the side. And then you're going to grab a colony of bacteria from the plate. Look for a single colony that you can just gently scoop up, if you will, with your loop. Return the lid to the plate. Get your sterile TSA slant. You're going to remove the cap. You're going to flame the mouth. Doesn't show that here, but you got to do that. Then you're going to go in with your inoculating loop, and in the same way that you did the slant-to-slant transfer, you're going to go toward the bottom of the slant here, and you're going to do a zigzag pattern toward the surface of the slant, toward the mouth of the tube. Pass that through the flame a couple times before you recap it, and then of course sterilize your loop. You've just done a plate to slant transfer using aseptic technique. So that's pretty much exercise 8. Those tubes and those plates, of course, need to go into the incubator before you leave. Make sure you invert the plate. I'm sorry, not the plate. You're not doing any plates yet. Make sure you have all those test tubes labeled. Okay, your broth and your 2 slants. We're going to be doing a plate in exercise 9. Got ahead of myself a little bit. So let's go on to exercise 9, which is entitled Pure Culture Techniques. You're going to want to make sure you read over the narrative there. It talks about Robert Koch and his contributions to not only the development of Koch's postulates, but also how he was able to grow bacteria in pure culture. So when you're given a mixed culture, you can isolate those various bacteria such that you have separate colonies, right? We've talked about that, and the tools of the microbiology laboratory, chapter 3, I think, in the textbook, discusses all this too. So the first thing we're going to do in exercise 9 is a streak plate. I'm going to reshare that Word document that I referenced a little bit earlier. And there is a Zoom link that I want you to watch carefully. Watch this Zoom. I'm not Zoom link, it's a YouTube link. It's also in the Zoom lecture, by the way, in chapter 3. That's where I pulled this. I want you to watch this YouTube video. It talks about one method you can use to do a streak plate. There are a lot of different possible streak plate methods. On page 74 of your book, figure 9.2, they describe 3 different streak patterns that you can use to obtain isolated colonies. Remember, the goal here is achieving isolated colonies. So again, watch the video, use their method to a T, follow what they talk about, and that's the method I want you to use. So what you're going to be doing is you're going to be using one of the, I think Deb has a mixed broth, and she'll have a test tube labeled exercise 9. It says E. coli and Serratia marcescens. The tubes will be labeled. They're TSB, E. coli, Serratia. So it's got 2 bacteria in that broth. Your goal is to separate those out in the streak plate method such that you can identify separate isolated colonies, right? So you're going to follow the methodology that we talked about in exercise 8 with regard to first mixing the E. coli Serratia tube, right? Mix it between the palms of your hands. You're going to sterilize your loop. You're going to remove some inoculum from the mixed broth, making sure that you are flaming the mouth before going in and all that stuff. I'm assuming now you know how to do all that. And then you're going to, again, follow to a T the description described in the YouTube video. Okay, so please follow those steps. You will then be labeling the bottom of your plate, right, with your name, the date. You've got both E. coli and Serratia. Okay, so make sure you do that labeling. And then eventually that will be inverted, right? You'll put the plate into the incubator with the lid facing down. And we talked about why we do it that way. Hope you remember why. The last part of exercise 9 is going to involve doing what's referred to as the pour plate method. So they talk a little bit about pouring an agar plate for streaking. We're not going to do that. Deb is going to be giving you a plate to streak, all right? So we will be not doing what's described in exercise 9, figure 9.3. Okay, just FYI. But I guess I would read over that so you have some idea of how a plate is poured. But we're going to be talking about the pour plate method first, starting there on page 78 of the lab book. And what I'm going to do is have you look again at this diagram where I have in the box pour plate method you'll be doing. At those 3 lab benches, there will be some warm water baths that Deb has set up. And I think it says 50 degrees on the digital readout of those 3 warm water baths. Inside each bath will be a test tube rack containing liquid trypticase soy agar tubes. Each one of those tubes has enough agar in it to pour 1 plate. So each of you is going to be using 3 tubes. 1 tube will be called tube number 1, there'll be a tube number 2, and you need to designate a tube number 3. I don't care whether you write that number on the actual tubes in the baths, or whether you just remember the left hand of the 3 is 1, the middle one is 2, and the right hand third one, is number 3. Just make sure you know what tube is 1, what tube is 2, what tube is 3. Now those are kept at 50 degrees because at 42 Celsius, molten liquid agar will begin to gel as it cools. 42 degrees is the magic number. So if we keep it above 42, and of course 50 is perfect, it'll stay nice and liquid or molten. But as it cools toward 42 and below, it will gel up kind of like Jell-O does, right? When it begins to cool, it sort of solidifies. Same idea. So what you're going to do, each of you will do this separately, is you'll get that mixed broth that you used for the streak plate. That was the E. coli serratia broth that Deb has labeled. And you're going to mix the tube. You're going to aseptically go in, get some inoculum, following aseptic techniques, right? And then you're going to place a loop full of mixed serratia and E. coli into tube number 1. And that's what's shown here on the handout. Now remember, these are going to have caps on them. They're not going to be open. So you need to make sure you remove the cap, you flame the mouth of the molten agar before you inoculate it, and then you flame it again before you recap it. So do that, inoculate the first tube, mix it between the palms of your hands at some point in time. I would do that after you sterilize the flame, or after you sterilize the loop. So inoculate it, recap it, put it back in the warm water bath, sterilize your loop, okay, and then go back in and pull it out and then mix. I would do that because if you mix it right away, you've got a contaminated loop to deal with, which could be flailing all over the place as you're mixing and all that. So inoculate the tube, put it in the warm water bath, sterilize your loop, then go back and grab that tube and then mix it really well. The reason we want to mix it really well, and it's really critical you do that, is because there could be clumps of cells, mixed E. coli and serratia together. By mixing it, we're dispersing the cells so that they're going to hopefully be floating out there as single cells. So you've got to make sure you mix it well at the end. Okay, then what you're going to do is you're going to aseptically transfer a loop full of contents from tube 1 into tube number 2. Okay, so again, following good aseptic technique, make that transfer from tube 1 to tube 2. This tube needs to be mixed really, really well at some point in time before you transfer your last loopful from 2 into 3. Make sure you flame the loops here between transfers. You will be left with 3 tubes of molten tryptic soy agar. Tube number 1, tube number 2, and tube number 3, each of which has bacteria in it, presumably, with tube 1 having more cells than 2, than 3, hence the difference in coloration here. Now you will be taking those 3 separate tubes and you'll be pouring them into 3 separately labeled sterile plates. So there'll be a stack of plates back there somewhere where you can just get 3 unused sterile plates, and you're going to label the bottom of the plate with your name, the date, mixed culture, all that information. So make sure you label these. One's going to be plate 1, plate 2, plate 3, coming from the 3 tubes. Make sure that you pour the bottom of the plate. Now you're going to have labeled the bottom of the plate. You got to take the plate and turn it upside down when you go to pour it. Otherwise you'll be pouring the lid. Don't pour the lid of the plate. Label the underside of the plate, flip it over after you've labeled all 3, and then you're going to aseptically transfer the contents. Now remember I said earlier, Deb has measured the volume of these tubes such that each will pour a plate. When you go to pour the plate, Just lift the lid off the top a little bit, pour the entire contents of the plate in one fell swoop, just gently, but don't dilly-dally, pour it all in. Put the lid of the plate back, and then gently, gently swirl the plate so that the entire bottom of the plate is covered with agar, molten agar. If you just gently rotate it, you'll disperse the molten agar such that it forms a small little covering on the bottom of the plate. Let those plates sit there for at least 15 minutes. And as they cool, of course, they will solidify. And then when the time comes, you can take them, invert them, and incubate them. Okay. So that's basically the pour plate method. The idea is when we go to pull these plates out and look at them on Friday or whenever you come in and take a look at these. And by the way, somebody should be planning to pull these tubes from exercise 8 and the plates from exercise 9. I would say Friday. I think, are you guys at school, I think, for nursing lab, some of you? I'm going to just ask that you coordinate your efforts here somehow. We don't want those plates in there for a whole week, or tubes. They got to be pulled, assessed, and put in the fridge. And then we can talk about them when we get together that following Tuesday. Exercise 18, entitled Culture Media Preparation. I took the students into the prep room and I showed them sort of how the agar comes in the powdered form and how it's mixed with water and then it's dissolved. And I talked about the autoclave and the fact that you have to have, you know, the temperature up to 121 Celsius at 15 psi, pounds per square inch, for 15 to 20 minutes to ensure sterility. And Deb was back there and she graciously kind of talked about kind of how she prepares the plates and the tubes. So it was very interesting, and I'm sorry you guys missed that. Maybe you know, we can take some time at some point and I can take you over there and kind of show you some stuff too. But I'd like you to read over this exercise. It talks a little bit about energy, carbon sources, nutritional requirements of bacteria, which we'll be getting into in lecture in another chapter coming up. It also talks about the difference between complex and defined media. I talked about that recently in the Zoom PowerPoint for lecture as well, so that's not new. And finally, the difference between selective and differential media. They talk about that there on page 130. So please make sure you understand the difference between those two. And then, of course, they also get into the autoclave and those various parameters that must be met before we can assume sterile conditions. We've killed all those endospores and so forth. So I'd like you to read over that exercise 18 as I mentioned in the syllabus. Okay, I hope this little Zoom recording was helpful to you. I am under the assumption that Wednesday, once we come back from the President's Day break, which is the 17th of February, is a good day for you to get in there and get those exercises made up. I'm sorry you had to quarantine. I hope you're all feeling well and not sick. But Deb will have everything out for you on Wednesday. And if you can get that done, then it'll just be a little hiccup in our schedule. We'll get back on track, I hope. If you have any questions, feel free to email me. And until we meet again, you have a great President's Day weekend.